All right, so welcome to XenoTech's webinar, Red Blood Cell Partitioning Studies to Improve Accuracy in Pharmacokinetics Calculations. Today I'm joined by subject matter experts Steve McGreal and Pallavi Lemaye to discover considerations and impacts pertaining to red blood cell partitioning studies as part of a preclinical drug safety assessment. Subject matter expert Dr. Steve McGreal will be delivering the keynote presentation, and then he will be joined by Director of Scientific Consulting, Dr. Pallavi Lemaye, for the question and answer session. Dr. McGreal is a research scientist in the Contract Services Division, serving as study director for non-clinical studies, including drug metabolism, enzyme induction, and enzyme inhibition. He's also responsible for review and interpretation of data, communicating results to sponsors, and responding to quality assurance audits. Dr. McGreal is a published author on 6 peer-reviewed studies and is trained to meet international regulatory compliance standards. He received his Ph.D. in toxicology from the University of Kansas Medical Center in 2017, and later that year joined XenoTech. Dr. Pallavi Lemaye is a director in the Scientific Consulting Department, providing input on drug-drug interaction study needs for sponsors. She's a published author of many peer-reviewed original research publications and several book chapters in the fields of toxicology and liver pathobiology. Dr. Lemaye currently serves on the program committee of the American College of Toxicology and is the current president-elect of the Central States Regional Chapter of the Society of Toxicology. She received her Ph.D. in toxicology from the University of Louisiana, and since then has cultivated a broad range of experience and expertise in IND-enabling studies. With that, I'll hand it over to Steve for today's presentation.
Thank you, Madison, for the wonderful introduction. So we're going to talk today a little bit about red blood cell partitioning, what it is, and how it can be useful for you for your compounds. Anyway, we'll start with the presentation outline, where we'll talk about what red blood cell partitioning is, why measuring it is important, and then we'll go into some of our partitioning study designs here at XenoTech, and then I'll show some data examples from our qualification study. To begin, I think we'll start with an interactive web question for the audience. So if you've conducted an RBC partitioning study before, was there a particular trigger for it? So, all right, well, thanks everyone for participating in our first interactive poll. So we'll start our talk today with just a basic overview of the composition of blood. So the average adult has about 5 to 6 liters of blood in their system, depending on body mass. Red blood cells occupy about 45% of the total volume of blood, and about 99% of the cellular volume. The remaining 55% approximately of your blood is made up of plasma, and then about 1% is white blood cells or platelets. And as I mentioned, what kind of makes this red blood cell partitioning pretty important, so you can see there's a pretty large percentage of the blood that's made up of red blood cells, and like I said, it accounts for about 99% of the total cellular volume of your blood. So what is red blood cell partitioning? Well, put simply, certain compounds will preferentially sequester into red blood cells over the plasma. We're able to measure this disparity by looking at the blood-to-plasma ratio, where we can determine the amount of compound that goes into each compartment of the blood. Knowing the blood-to-plasma ratio, this RB, is an important parameter that can help determine whole blood pharmacokinetics. And the failure to take into account the red blood cell partitioning can lead to an overestimation of intrinsic clearance. And I've got a few examples coming up here in a few minutes that will kind of illustrate that point and hopefully make it a little bit more clear. So another reason why measuring RBC partitioning is important, if you look at this maximum unbound hepatic inlet concentration, this is what we're talking about right here. It's part of the equation. So measuring the blood to plasma ratio is important for finding out, basically the hepatic inlet concentrations of certain compounds and drugs. So from that sense, it's a pretty invaluable and important tool to help with some of these concentration determinations and some of the pharmacokinetics and things like that. So it's a very important parameter to have measured. So traditionally, pharmacokinetic parameters have been measured using plasma instead of whole blood. And as I mentioned previously, certain compounds do have a really high affinity for red blood cells. So they'll have a pretty large blood to plasma ratio. And this can lead to an overestimation of your blood clearance if you only analyze the plasma. So for these compounds with the high ratios, the concentration in the whole blood in a lot of cases can be a more accurate parameter to use for your pharmacokinetic studies. As I mentioned, if you don't take it into account and you do have a really high blood to plasma ratio, you can definitely overestimate your blood clearance. So the effects of red blood cell partitioning. So compounds can enter red blood cells in several different ways. A lot of lipophilic organic compounds will dissolve in the lipid bilayer of the membrane and enter the red blood cells. Small hydrophilic compounds can enter by diffusion. And just as a general rule, when we're talking about blood to plasma ratios, non-ionized or basic drugs typically have a blood to plasma ratio close to 1, meaning that they will distribute evenly between the blood and the plasma. In contrast, acidic or deuterionic drugs are excluded from the erythrocytes, and so therefore they'll have a blood to plasma ratio of roughly 0.55. So they preferentially stay in the plasma compartment of the blood. The red blood cell partitioning assay can give us a method to determine the extent to which a compound is sequestered in the red blood cells. Because like I said, it's in the name, we measure both the blood concentration and the plasma concentration to determine the ratio. So a lot of compounds will have a really high blood to plasma ratio. A good example is chloroquine. We actually use chloroquine as our positive control, and chloroquine's been shown to have a blood to plasma ratio of greater than 2. So it is preferentially sequestered into the red blood cells. Other compounds, such as metoprolol, will have a high affinity for the plasma, and they'll have a high affinity to the plasma or equal affinity to the plasma of the blood. And those will typically have a blood to plasma ratio of less than 1. So we've used metoprolol as our basically as our negative control, so it doesn't show preference for the blood or the plasma, and its blood to plasma ratio is about 1. So as I mentioned, there's a lot of variance between different drugs and compounds with their blood to plasma ratio. Darepinum is one that has a blood to plasma value of about 0.5, which means that it's highly distributed to the plasma. So taking this into account, if we were to calculate the exposure using the plasma, we're going to have a value that's almost twice as high as if we use the value of exposure for the blood. On the other side of that, we have butorphanol, which has a blood to plasma ratio of about 2. So it's highly distributed into the blood. So if we were to calculate exposure using the blood, it'll be almost twice that exposure of the plasma. So you can kind of get a sense from those two examples. The pretty widely distributed blood to plasma ratios, 1 of 0.5 and 1 of 0.2, can give you vastly different calculated effects depending on where the drug falls on that blood to plasma ratio. So it is very important to take into account depending on your circumstance. All right, we'll talk a little bit about tacrolimus. It's highly bound to the red blood cells. So it's got a blood to plasma ratio of about 15, and its volume of distribution and hepatic clearance vary greatly depending on whether you look at the blood or the plasma concentrations. So if we take a look at the volumes of distribution and we look at the blood, we've got about 1 to 1.5 liters per kilogram. But if you look at the plasma, we've got about 30 liters per kilogram of the volume of distribution. If we look at the clearance for blood, it's between 2.1 and 6.3 liters per hour. But if we look at the plasma, you can see we've got 42 to 378 liters per hour. So if we use the plasma concentration, the clearance actually exceeds the hepatic blood flow and the hepatic plasma flow. The hepatic blood flow is about 97 liters per hour, and the hepatic plasma flow is about 53 liters per hour. So you can see if we were to use the plasma to calculate our clearance or our volume of distribution in this case, we would have values that were much higher than if we had used the whole blood. So the plasma definitely overestimates both the volume distribution and the hepatic clearance. So while that's an important, while blood to plasma ratio is an important thing to consider, when talking about distributions and clearance. There are certain drugs that are highly bound to red blood cells, and this can affect their ability to move in and out of the red blood cells. So certain diuretic drugs like chlorthalidone and dorzolamide, they'll bind with really high affinity to carbonic anhydrases inside the red blood cells. And due to this binding, they don't move freely from the erythrocytes back to the plasma. So these compounds, while they do have a really high blood to plasma ratio, it ranges from about 30 to 240. In these cases, using the concentration in the plasma is more relevant to their ability to cause drug-to-drug interactions, because they do bind so preferentially to carbonic anhydrase that once they bind inside the erythrocytes, they're not getting back out into the plasma. So in these cases, while we do have a high blood to plasma ratio, it does make more sense to use the plasma to calculate these concentrations. Use the plasma to calculate our parameters because it is a little bit more accurate, since we're not getting back out of the red blood cells once it's entered. All right, that brings us to our second interactive webinar question. When in the developmental process do you conduct red blood cell partitioning studies? That's good info, and I want to thank everybody again for participating in our poll. So what knowledge can be gained from these red blood cell partitioning studies? Well, I mean, I think first and foremost, it can give you a rational choice for your appropriate biological fluid. As I mentioned previously, a lot of pharmacokinetic parameters have traditionally been calculated using the plasma. However, if you do have a drug that's really preferentially sequestered into the red blood cells, it could make more sense to use whole blood instead of plasma for your downstream assays. It also gives you a physiologically meaningful reference for your parameters of the drug concentration in either the whole blood, the plasma, or the serum. And it's also an effective screening tool for drugs whose biophase reside within the RBCs. And so it enables the study of the effects of drugs on red blood cells. That brings us to our next section, where we kind of talk about the Sekisui XenoTech's approach and study design for red blood cell partitioning. So our studies, we use whole blood, and we use the anticoagulant K2-EDTA. Our hallmark is these 5 species, so mouse, rat, dog, monkey, and human. For mouse, rat, dog, and monkey, we typically use pools of 3 or more, exclusively male. For humans, we use pool of 3 or more, mixed gender. We do use fresh blood, so we have a local vendor that's able to provide us fresh mouse, rat, dog, and monkey blood, so we're able to get it the same day. And then typically, if we have a study that requires human blood, we'll contract with a phlebotomist to come in and collect blood from our employee volunteers. We typically do 3 test concentrations. All of our incubations occur at 37 degrees C for two time points, obviously 0 hour, and then we've been going out up to 60 minutes. But if you have some special information that you already know about your drug, like, you know, if it's rapidly equilibrized into the red blood cells or something like that, we can cut down the time. That's not really an issue. We use a high affinity control. Chloroquine, as I mentioned previously, has a pretty high blood to plasma ratio. And then we use a low affinity control, metoprolol, just to prove competency of our test system. As I mentioned previously, metoprolol has a red blood to plasma ratio of around 1. And chloroquine has a blood to plasma ratio typically over 2. So we will basically take an aliquot of whole blood, and we'll take it out for our whole blood fraction. Then we'll centrifuge the rest and extract the plasma. At the end of each time point, the whole blood and isolated plasma will mix with stop reagent, and we will run that both the whole blood and the isolated plasma samples on LCMS, and that way we can estimate the aniline internal standard area ratios. So we've not been using calibration curves. We found we get really good results just using area ratio. We look for 3 parameters. Our first parameter that we look at is stability. And this is basically the concentration of the incubated blood over the concentration of the 0-minute blood. And we look at this mainly to make sure that there's no degradation of the test article or compound over the course of our assay. The second thing we'll look for is the distribution into the blood cells. And this is expressed as a percentage. So it gets reported as a percentage of the compound that sequesters in the red blood cells. And then the last thing we look at is the blood to plasma ratio, and that's basically the concentration in the whole blood over the concentration in the plasma. And here's some of the results from our qualification studies. As I mentioned previously, we did mouse, rat, dog, monkey, and human. You can see in this first column right here that we have really good stability over the time course of our assay. So over our 60 minutes, we're looking at somewhere between 89 and 100% stability, which means that our compound chloroquine is not being degraded over the course of the assay. And then you can see the distribution, about 80% to about 90% distribution into the blood cells. And this gives a blood to plasma ratio anywhere from 2.8 up to about 6.2 for dog. And for human, actually, we've got about 3.06 blood to plasma ratio, which is right in line with literature values. So the literature values that I found were between like 3.2 and 3.5. And we're looking at about 3.06 with a variance of about 0.45. So we are right in that comparison range for our values versus the literature values. We also looked at the low affinity control, metoprolol, same species as we did for chloroquine. You can see here that the stability for metoprolol is also very good, between about 90 and 100%. It distributes pretty evenly between about 44 and 53% to the blood, so it's about evenly distributed between the blood and the plasma, and that gives us a blood to plasma ratio of roughly 1. So for humans down here, we're at about 1.07 with a variance of about 0.05. If we look at some literature values, about 1.1, 1.2 for metoprolol. And as you can see, we're right in that range. So our assay compares pretty well with the established literature values for blood to plasma ratio. So the next slide is just a summary kind of what we talked about. I think the main take-home point from this talk is that certain compounds are preferentially sequestered into the red blood cells over the plasma. And this makes using whole blood a more appropriate parameter for calculating downrange pharmacokinetic parameters, because if you're using just the plasma, you can overestimate clearance and volume of distribution and things like that. So for instances where you do have a really high preference for the red blood cells, it makes sense to use whole blood over plasma. So that brings us to our third interactive webinar question. After what you've learned during this webinar, would you be more likely to conduct RBC partitioning at the same time as plasma protein binding? It is kind of, I guess, two sides to the same coin with the red blood cell partitioning. If the blood's 55% plasma and 45% blood, it makes sense kind of to look at both of those parameters. All right. And that concludes the slide presentation portion of the seminar, and we'd be happy to take any questions that you have at this time. Thank you for listening.
Thank you so much, Steve. So while we wait for questions, I'd like to go ahead and take this opportunity to mention that information about our red blood cell partitioning and other contract services are available on our website. We also provide test systems for in-house assays and scientific resources relating to DDI and ADME, including published papers, posters, other webinars, and more, which are available on our website. If you have any further questions regarding today's presentation or any of Sekisui XenoTech's contract research services or products, please feel free to ask them in the post-webinar survey, or you can contact us through our website. And if you'd like to be notified of future webinars, you can submit a webinar topic request form or register for our newsletter.
I've got the first question here. Can we segregate drugs inside the red blood cells? Bind to them? Do they bind to the membrane? Are they chemically bound or just attached? So as far as the assay goes, we cannot distinguish between whether a drug is chemically bound or if it just is concentrationally driven across the membrane of the blood cells. But in general, yeah, all of those cases can occur. So as we mentioned previously, some compounds will simply diffuse across. Other compounds, it's temperature or pH driven. The pH of the blood is roughly 7.4, but the pH of the actual cell volume inside the blood, the red blood cells, is a little bit lower, like around 7.1, 7.2, in that range. So from that perspective, all of these parameters kind of play a role into whether or not your drug is going to migrate into the cell volume or stay in the plasma. But as far as the assay goes, we're not able to distinguish between whether it's a carbonic anhydrase binder, whether it gets trapped in the membrane, or whether it just simply diffuses across and then moves back across the membrane.
All right. So the next question is, what are the cases that you should use blood concentration for DDI estimate? So if you look at the guidance by either FDA, EMA, or PMDA, all three regulatory agencies do require or recommend that you do the basic and static model in order to figure out if there is a clinical DDI potential. And if you look at those equations, all those three equations do require the RB, or the blood to plasma ratio, RBC partitioning ratio, in that calculation. One of the slides that Steve showed did show that equation where that RB factor will be incorporated, that is for the inlet concentration of the drug. And all three agencies do require that. There are certain slight differences in those equations, but ultimately they are wanting us to use the systemic concentration, which is Cmax, as well as the portal concentration, or the inlet concentration of the drug in the hepatocyte. So that's where that has been used. Unless you know that the drug is preferentially bound, like what Steve mentioned, inside the RBCs to certain enzymes, then you know that the drug is not freely moving back into the plasma. In that case situations, you would not use the RB factor.
So there was another question about whether or not we can use simulation programs to predict the red blood to plasma ratio. So there is a really nice paper about it. However, I think the results are somewhat inconclusive, just because it's kind of difficult to predict the thing. But if you like, I can forward you on a link to the paper. That way I don't underrepresent or overrepresent their findings. Okay, so the next question is, can you comment on total whole blood concentration versus the free drug hypothesis? Yeah, so in order for the drug to be available to enter the red blood cells, it obviously needs to not be bound to any plasma proteins. So, and in that instance, only unbound drug in the blood will enter the red blood cells. Next question, how long can you run the blood incubation assay? A lot of these compounds, especially some of the hallmark compounds that we use as controls, are pretty rapidly equilibrized. So you see partitioning relatively early on. But we can probably go out to a little bit longer time points. I think my only real concern with that is, since we are using the fresh blood, I might be a little concerned with some hemolysis being outside of the body. But yeah, if you've got a request for a longer time point, that's something that we could definitely discuss.
All right. So the next question is, what concentration range do you recommend using? So in that, if you know your clinical dose, we do recommend that you bracket that dose in your 3 concentrations that you will be using.
What is the significance of the percent distribution? So the percent distribution is basically it's used to kind of, we give the blood to plasma ratio, which, for some of these compounds, let's just take chloroquine, for example, in humans, it's about 3. But the percent distribution basically it takes into account the hematocrit volume as well. So we're able to basically adjust that distribution for the different compartments. And so it gives you a percentage for how much of that drug was found in either the blood, the whole blood, or the plasma. So in cases of chloroquine, I think we're seeing upwards of 80%. It means once we've isolated the blood or spun down the whole blood and we've got isolated cells and plasma, we're seeing about 80% of the drug is staying in the whole blood fraction and is not in about, the opposite of that would be about 20% of the drug is still in the plasma. Next question, kind of related. Yes, we do measure the hematocrit for each experiment. That's something that we measure right away. So that's a great question.
So how often do you see species difference in the blood to plasma ratio? And that really depends on the drug that we are looking at. So we would recommend that for your toxicology species, knowing the difference in that blood to plasma partitioning will be a good idea. That way you can relate to the PK parameters or the results that you have obtained in the animal studies are true representative of the human situation. Another question is, how does hemolysis impact the assay calculations? I would say that is definitely a concern, because if you have hemolysis, then it's sort of contamination, and you have already accounting for some of the drug concentrations from the RBCs. So we would be, when we do the assay, we definitely watch for any hemolysis when we centrifuge and collect plasma.
Yeah, that's right. We did some experiments in our qualification study where we used stored blood, and we didn't see as well of a separation between the blood and the plasma, and we didn't see as higher ratios either when we looked at the blood to plasma ratio. And it was mostly attributed to the fact that over time we just get hemolysis. So as we get less and less cell volume, we obviously will get less and less blood to plasma ratio. So here's a question about dose response or dose range in the study. So yeah, we typically, our typical design is to look at 3 different concentrations. And it has been shown that certain compounds do have concentration dependence to their red blood cell partitioning. So yeah, I think as we mentioned previously, with the choosing the dose, we would kind of want to bracket our target dose, and just to make sure that we're kind of covering our bases. But yeah, with the 3 concentration levels, while it's not a true dose response, because we obviously would need to add a few more concentrations in, we are able to see if there's any sort of pattern between a lower dose, our target dose, and the higher dose.
Right, but there is a possibility that you may see dose-dependent changes in the RB values, and mainly this partitioning is via diffusion. But there is a possibility that some of the carriers and channels are also partitioning the drug, and in that case, you will see a saturation, and that is the reason why you see the dose-dependent changes in the RB value.
Here's a question about radioactive molecules. We do not use the radioactive molecules in the Kansas location. However, our colleagues in Japan, they do use radioactive monitoring when they do the red blood cell partitioning. And then also the next question right below it, do we work with larger molecules for the RBC assays? I think a lot of this would kind of depend on, a lot of the larger molecules kind of depend on, I guess, kind of whether they're soluble or not in the blood. So we could look at larger molecules, yes. Here's one about the fraction unbound for drugs with a high RB. The fraction unbound in the plasma, you can, if you know the blood to plasma ratio, you can interconvert between the fraction unbound in the plasma and the fraction unbound in the actual blood. Here's one about the importance of using fresh blood. I don't know. I know in our experience, we've got a lot better results using fresh blood. And also another factor I think that plays into it is the time it takes for delivery. So I'm not sure that we would necessarily see hugely drastically different results if we use store blood, but I've always kind of, when we order, so we get to have a local vendor and they're able to actually like have a courier deliver the blood. So the time from when they draw the blood till the time when we run the assay is typically a couple of hours. We've ordered blood from several online vendors before. We do see similar results, but not as dramatic. So it just kind of, the fresh blood we've always felt like gave us better results. So we kind of lean towards using that fresh blood when we can.
Again, what is the current understanding around RBC transporters that may affect partitioning? So it's mainly the carriers and the channels that participate in this RBC partitioning of the drugs. In vitro blood to plasma versus in vivo blood to plasma, can you comment if they can be different? So in literature, there have been some reports where they did see a difference in vitro versus the in vivo. And that was partly because of the pH changes and some other factors, but that is a possibility that we have found in literature. And if you're interested, I can send you that link to that paper that talks about this difference. Does in vitro binding carbonic anhydrase assay predictive of RBC partitioning? It is a possibility that if it is a carbonic anhydrase inhibitor and it binds, then it will be a predictive assay for RBC partitioning.
Here's one about the physiochemical properties of the drug. Will it affect the partitioning? Yeah, as we kind of talked about previously, there's some general rules on different compounds for whether or not they will partition into the blood or not. So yeah, absolutely. Any physiochemical properties surrounding the drug can potentially affect what we're looking at. Here's another one. What if the ratio is just below 1, so around 0.7 to 0.9? What would you say about the diffusion? So in cases where we are right around 1, so I think from that point, since we are looking at the whole blood to plasma, anything below 1, we are probably going to say that it's not preferentially sequestering the blood. And also, since we are using whole blood, most of the values that we'll report will probably be right within that range. So I think we would probably say that it's just not, if it's under 1, it's not going to be partitioned into the red blood cells at all, since we are not measuring, we're measuring whole blood to plasma as opposed to reconstituted red blood cells versus the plasma. It can be calculated that way as well, but we typically use the whole blood instead.
Okay, so there's one more question. Any idea if large compounds such as beyond the rule of 5 compounds have a preference for plasma versus RBCs? And really, I mean, this rule of 5, which means that the drugs that have not more than 5 hydrogen bonds as donors, 10 hydrogen bonds as acceptors, and log P value of 5 and lower. We have not come across many examples of that. There is one paper that I did find that talks about the Protac drug, which is against c-Myc. And in that study, they did find that this drug was more in the plasma and not partitioning so much in the RBCs. So that is sort of the only paper that I have found so far. And again, I can send you the link to that paper for your reference.
Here's another question about, do we have experience with compounds that are metabolized by the blood enzymes? So in this case, if they are being metabolized, we're likely to underreport our blood to plasma ratio, since the drug is entering the red blood cells. But we're just basically, at that point, we wouldn't be collecting all of the parent drug when we're analyzing it by mass spec. I'm trying to think if I haven't had any, I haven't seen anything like that, but I guess a sign would be that our recovery at 60 minutes in the whole blood sample is much lower. Also in the plasma sample, if we start to see like a loss, I mean, it would also be able to tell that from the stability data, because when we look at stability, we're basically looking to see if there's any either degradation or metabolism of that drug over our time course. So in that instance, if we're seeing a pretty low stability, I think we lend you to think that potentially we're seeing some kind of metabolism.
There's another question. Just share it. Sorry, it was about the carbonic anhydrase inhibitors and why we are calling them irreversible. So it is really depending on the property of the drug. If we know that it is irreversible, for example, diuretic drugs, which we know that those are irreversible, and in that case, we know that they will not be moving back into the plasma that easily.
Here's a question about dose dependence. It has been shown that there are several compounds that are dose dependent. Like I mentioned previously, there's a lot of factors that kind of play into why a drug might be partitioned into the red blood cells. I've mentioned concentration. pH is another one. Binding, it would be one. Like I said, certain compounds are temperature sensitive, so they will partition at physiological pH, but they will not partition at room temperature. So yeah, there's a lot of those things that kind of play in to why we'll get compounds that have a high blood to plasma ratio.
So the next question is, will the RB change with disease? Yes, there are some references in the literature that do show that RB can change with disease. Like malaria is a good example, where if you have the plasmodium infection, the RB value with and without is going to change.
Here's an interesting question. Is there a difference between whole blood partitioning and red blood partitioning? So when we talk about the partitioning and the RBC partitioning that we've talked about today, we're only looking at the whole blood fraction versus the plasma fraction. So you can look at actually reconstituted red blood cells as well to find out exactly how much partitioning has occurred. But when we talk about it, we're strictly looking at blood versus plasma, and we're kind of using it as kind of a stand-in. But yeah, you can actually separate them both out, reconstitute the red blood cells, and look at the fraction in the red blood cells. But we typically look at the value of the plasma versus the whole blood to look at our partitioning. I'll switch this one from Nadia. Is it important to assess stability of the compound in the whole blood? Yes. I think we monitor it because we want to know that we don't that we're not seeing degradation. Or so I mean, I think it's kind of important because, you know, if the stability drops to, say, you know, 50% or whatever, it could bias our results, because we would see an artificial change in our blood to plasma ratios and in our distribution, which could likely not be due to the partitioning. So we kind of monitor the stability to make sure that over the time course of the experiment. So we're typically working with an hour or less for some of these experiments. So we just want to make sure that we're not seeing any degradation of the compound in the blood, so we can say the results that we're reporting are accurate.
Okay, great. Thank you so much to Stephen Pilevi. So that wraps up today's session. Please stick around for the post-webinar survey. It's quite quick. Thanks to everybody for taking time out of your day to join us. And a big thank you to Stephen Pilevi for that great presentation. Everybody have a great rest of your day.
Yeah, thanks everybody for listening.
Thank you. Thank you.